My name is Srivastava, and I'm presenting on gene therapy for ocular diseases. I'm a current junior at Independence High School, which is in Frisco, Texas. Here are some of my extracurriculars that I'm currently in. Eventually, I hope to pursue a career in medicine or neuroscience. So, starting off with a little statistics about ocular health on a worldwide scale. Out of the 2.2 billion people with some form of vision impairment, 45% of them are yet to be addressed and see some form of proper treatment, leaving them to live their daily lives with a number of proven social and cognitive disadvantages, causing $411 billion to be lost in economic productivity. There's also a shocking inequality across the world, as lower and middle income countries have a 4 times greater prevalence of vision impairment per capita as compared to higher income countries. Let's go over some basic anatomy of common diseases. So the cornea and the lens work to focus and refract light rays onto the retina, which contains photoreceptor cells that process the light and send information to the brain through neurons in the optic nerve. From presbyopia, the loss of nearby vision due to aging of the lens, to diabetic retinopathy, which inflames the blood vessels in the optic nerve and can eventually cause complete blindness. These disease causes can be anything ranging from external bacteria, viral infections, vitamin deficiencies, general aging, as well as genetics and gene mutations, which we'll be focusing on. Treatments today often can be risky due to the eye's fragile state, sometimes with not too high success rates. Now, gene therapy aims to provide hope as an emerging solution. So, what is gene therapy? Simply, gene therapy works to alter genetic code to restore the proper functions of essential proteins, usually when a harmful mutation has occurred. Gene replacement therapy involves introducing a normal copy of a gene to replace a defective one in cells, restoring normal function by compensating for the faulty gene. Gene silencing techniques, such as RNA interference, known as RNAi, degrade or prevent the translation of the gene's mRNA, which converts the DNA information to synthesized proteins. These methods can effectively silence harmful genes, providing therapeutic benefits conditions caused by the overexpression or aberrant activity of certain genes. Gene editing, on the other hand, exemplified by CRISPR-Cas9 technology, allows precise modifications to an organism's DNA. By targeting specific DNA sequences, scientists can alter genetic material. Now, viral versus non-viral vector delivery. Viral vector delivery uses a modified virus structure, just replacing the actual virus DNA with a therapeutic gene to deliver the gene directly to the cell using the virus's built-in cell integration tools. AAVs, known as adeno-associated viruses, are the most commonly used virus for viral gene therapy, as they're more stable with typically less side effects. On the other hand, non-viral delivery provides many alternatives to latch into the cell membrane to facilitate entry into the cells as compared to the commonly used viral form, such as the ones listed below. There are also non-invasive and invasive administration methods. Non-invasive methods deliver therapeutic agents without requiring entry into the body or breaking the skin. This can include oral or topical administration. Invasive methods involve procedures that penetrate the body, often requiring medical intervention, including various types of injections and surgical implantation. Now that I've introduced gene therapy, why should it be utilized in place of other eye treatments? Firstly, gene therapy only requires one quick dose as compared to other treatments where the process may be time-taking, arduous, and possibly painful for the patient to endure. Specifically to the eye, though, gene therapy also provides a localized treatment and has practically no effect outside of the ocular region, making it safer for the rest of the body. The eye is also a relatively fragile organ, and opting for a simple gene delivery instead of indwelling devices or risky treatments like surgery reduces the chances for other issues or human errors. Gene therapy also boasts increasingly high success rates due to its high accuracy and the fact that it's customizable, meaning it can treat different patients across different genomic backgrounds. Now, let's analyze the look-starness process, an example of a current working gene replacement therapy that treats Leber congenital amaurosis, or retinal disease. So let's start with what normal eye function looks like in a normal visual cycle. The RPE65 enzymes converts all transretinal esters, which store essential vitamin A, to the chromophore 11 cisretinol, which absorb incoming light. 11 cisretinol then combines with the proteins known as opsins to provide necessary visual pigments known as rhodopsin in rod cells and photopsin in cone cells. This combination is essential for the absorption of light by photoreceptor cells in the retina, initiating the process of vision by processing light into electrical signals that are then transmitted to the brain for interpretation. When RPE65 is mutated, its deficiency causes less all-transretinal esters to be converted to 11 cis-retinol. Without sufficient 11 cis-retinol, photoreceptor cells cannot regenerate their visual pigments effectively. This causes LCA, Leber congenital amaurosis, a disease whose symptoms range from nyctalopia, which is night blindness, to peripheral vision loss and light sensitivity due to the photoreceptor cells not having enough pigment cells to process light effectively. Now gene therapy's role comes in. Luxturna is delivered via a subretinal injection and enters the cell through receptor-mediated endocytosis. This binding triggers the formation of a vesicle from the cell membrane, which enters the cell then sheds its outer clearance shell, allowing the therapeutic genes to enter the damaged cell. After receptor-mediated endocytosis, internalized ligands and receptors which are essentially chemical messengers, activate intracellular signaling pathways, leading to the activation of transcription factors. These factors enter the nucleus and bind to specific DNA sequences, initiating transcription. The resulting messenger RNA, mRNA, is processed and exported to the cytoplasm, where ribosomes translate it into polypeptides. During translation, transfer RNA molecules bring amino acids to the ribosome, matching their anticodons to the mRNA codons, thereby forming a polypeptide chain. The newly synthesized polypeptide undergoes folding and post-translational modifications to become a functional protein, replacing the previously damaged RPE65. As a result, the visual cycle can function properly, significantly improving symptoms of vision loss and sensitivity. Luxturna was revolutionary in that it provided for a disease that had no known cure before, improving eye test scores by about 9 times compared to people who didn't take Luxturna. However, there are some drawbacks to gene therapy methods. First is that gene therapy, from setting up infrastructure to dose administration, is exceptionally expensive, with costs reaching upwards of tens or even hundreds of thousands of dollars, including Luxturna's pricey $850,000 price tag, making it pretty much unaffordable for the average patient. However, it is predicted that these costs will be significantly reduced when the doses are produced in bulk on a larger scale, more efficiently, known as economies of scale. Additionally, the research on this is vastly uncovered, meaning scientists can't say much about the long-term effects of gene therapy and whether or not if it's a viable option. On another note, a question of ethics arises about whether or not using gene therapy is moral, as a lot of religious ideas say it goes against the natural given human body. Also, inheritable gene therapy, or gene therapy transfers to offspring, is banned in most areas, so advancements will be hard to make in that field. These concerns might likely slow the acceptance of gene therapy worldwide. As shown in the map, there is a shocking inequality between high-income or influential nations over lower and middle-income countries over their share in the gene therapy market. The US and UK alone share almost 70% of the market, while most developing nations share no more than a fraction of a percentage, highlighting the clear disparity in research infrastructure globally. So what can administrators do to make gene therapy more accessible worldwide? Firstly, increased research and infrastructure funding is needed, and this can happen with international collaboration and the help of governmental and non-governmental organizations like PEPFAR and the Gene Therapy Initiative. Secondly, a streamlined legal process, harmonized with international standards like those from the CMS, Centers for Medicare Services, accelerates gene therapy approvals, reduces costs, fosters innovation, and ensures patient safety, making advanced treatments more accessible globally. Changes in financial and market structures can greatly improve the success of gene therapy. Insurance and reimbursement policies that cover gene therapy can reduce costs for patients and encourage providers to use these treatments. Payment models like annuities, which spread costs over time, and other models may help advance the success of gene therapy from a financial perspective. Overall, as research continues to further build itself, gene therapy has hopes to be an influential cure for a growing number of ocular diseases. These promising benefits have led gene therapy to become one of the fastest growing areas of medical research, expected to see a market growth of almost 42 billion US dollars by 2033. Here are my references, and I would like to thank the GHLC team for giving me this opportunity, as well as my audience for watching. Thank you.